Now, welcome everyone who is here. We're here in person and everyone attending online, and also the ones who are watching the recorded video afterwards. It's my great pleasure to have most of the core of Genotropics here in Berlin today at the Leibniz Institute for Zoo and Wildlife Research. We are here in the lecture hall. I'm Camilla Mazzoni, a scientist here, group leader in conservation and evolutionary genomics. I'm here with my three colleagues. Three of us are going to present today, and Mariana Neri, Eduardo Ezrik, and myself. And at the end, we're going to have a discussion, which is going to take place mostly here in person. But we want to give the opportunity to also answer questions for everyone who is online. So we are going to probably present and have questions and answer for about 1 hour and 20 minutes. This can be extended in case there are many questions. And after that, we're going to stop the streaming and the Zoom call, and we're going to continue here in person. But for all of you who are there, you're very welcome. I hope that you enjoy, and feel free to ask your questions in the chat of YouTube at any moment, or any comments that you would like to do. Also visit our webpage, genotropics.org, online if you want to register in our contact list. And I'm going to just start here by briefly having my colleagues introducing themselves. And after that, Mariana is going to start with the first presentation. Mariana.
In order.
Okay, hello, I'm Mariana Neri. I'm an associate professor from the State University of Campinas, working mainly with genomics and evolution of aquatic mammals.
Hello, let me see. Okay, there. Am I showing?
Yeah, absolutely.
There. So I'm Eduardo Ezrik. I'm a professor at PUCRS, that's the Pontifical Catholic University of Rio Grande do Sul in southern Brazil. I'm also a member of the Genotropics consortium, and I mostly work on evolutionary and conservation genomics of carnivores.
Hi, everyone. My name is Ricardo Pereira. I'm a professor at the University of Hohenheim in Baden-Württemberg, and my department is based at the SMNS, the State Museum of Natural History in Stuttgart. And we study biodiversity monitoring at the genetic level, so how species can adapt and endure through environmental change.
Okay, so if you can give us just a heads up that you can hear us and everything, feel free to introduce yourself in the chat. I can see that some people are already doing that. And we are going to get started now with Mariana.
Right. See you soon, everyone.
All right. So good afternoon or good morning. I'm here to share an overview of our consortium, Genotropics, how the motivations, I'm listening myself. The motivations that led to its creation and some of the achievements from the last 2 years, basically setting the stage for the upcoming talks from Camilla and Eduardo Ezrik. So how it started, Genotropics was launched in 2023 through a joint program from Germany and Brazil program through the DAAD from the German side and CAPS from the Brazilian side. For us Brazilian, it's called a ProBrau program. It's a program that mainly funds exchange of PIs and students and some workshops. And that united 5 institutions by that time. By the beginning, it was, they already presented themselves. It was Unicamp, PUC-RS from Eduardo, UFMG from Fabrício, who is not here, and from the Germany side, Camilla, the Leibniz Institute that we are here today, and Ricardo, the Stuttgart National History Museum. And our main goal was to establish a consortium of experts to apply genomic approaches, studying neotropical species through training, knowledge transfer, and joint analysis of some sub-projects that we were already doing in our laboratories. All of us already worked with genomics So we joined efforts to make this first step, this next step, to form this consortium. And the consortium are structured in 3 pillars: the scientific articulation, the capacity building, and stakeholders interconnection, that I will go into more details, as long as I will be talking about our achievements in the last 2 years. As objectives that we had, and I will revisit them while I talk about what we did so far, was mainly to connect the experts in genomics working on neotropical species, to train students and young researchers in new genomic techniques, elaborate new joint proposals to expand the set of species, to analyze high-quality genomes using comparative and population genomics, to generate new genomic data sets, and to establish some protocols of legal and technical aspects to guarantee equity in the exchange of samples, genomic data, and researchers. Regarding the scientific articulation pillar, as I told you, when we started with these first sub-projects, focusing on the vertebrates that are shown there. It was collaborative projects that we were doing and are doing by that time and now, aiming to use these high-quality genomic data to understand evolutionary patterns, mainly specifically thinking about species adaptation in the Neotropics. Regarding that project, we are almost done or finishing most of them, and are currently doing and finishing some of them as well. Plan to finish it for the next year or so. Regarding the capacity building pillar, our idea was to use different resources to disseminate genome techniques, focusing mainly on young researchers, but using also different formats of workshop. For example, we had the workshop in 2023 in Belo Horizonte. It was a hybrid and focusing on genomic methods in non-model organisms, but I will show you as I speak about them. We had 60 participants from all over Brazil. It was an hybrid format, so we had persons in a room and also in the computer, and it was really great. The feedback from them, it was excellent, and we're mainly focused on the young. Following this workshop, we had the very first online symposium that almost marked our launch to the public. It was the first time that we were talking about the Genotropics to a big public, and we were surprised that reached a great interest. It was almost nearly 200 researchers studying neotropical diversity that were there in this online symposiums, together with, it was us 5, and together with some collaborators and friends that already work with genomics, or they are part of another consortium, like the GBB for the ETV, they are doing an excellent job producing genome from threatened species in Brazil. And it amazed us by that time how the level of interest that people showed. We didn't even know most of them, not most of them, but many of them. And we realized by that time the necessity really to connect all these people that were working individually in their laboratories, but were, eager to share, to be part of a consortium, to connect, because the challenges were the same about doing genomics in Brazil. So following that, we started a series of meetings and discussions that were very enriching for all of us, and mainly directed our next steps, as Camila will going to talk more soon. The workshop in 2024 was different from the year before because it was focused on researchers, not the youngest, the researchers, but more the more established researchers, the PIs, and we discussed how to do the collaborations that were equalitarian for the future. We discussed the challenge that we face, and we discussed the opportunities for the future. We discussed the interconnection with other consortiums and how we could, learn from them and where we would like to be going in the future. And we had, again, a huge success. All the videos from the 3 days that we were discussing it, they were there on YouTube, and we had more than 3,000 visualizations. Again, forecasting the interest of the people who works there, who works with genomics, in being part of this, in discussing all this. And this year, we have the how to say inscripções in English? The subscription applications are open for the workshop on population genomics. It will be a hands-on workshop. It will be held there in Porto Alegre, in the south of Brazil, but also will be in a hybrid format, so people can be part of it without the need to be in there. And it's going to happen in September this year. I think I can say that among our objectives, among our initial objectives, the first one that stands out was the connection with connecting experts in genomics working on neotropical species, that we really succeeded at that. We now have this Slack channel with almost 170 members. We have the mailing list of nearly 700 researchers and students in Brazil interested in the activities of the consortium. Following the symposium that I mentioned and the workshop, we have a joint workshop with the GBB, the other consortium that I mentioned for you in Brazil, and a symposium in the Brazilian genetics meeting with very interesting developments that led to the coordination with the Ministry of Science and Technology that is ongoing, so I cannot give much detail, but we are talking to them and they are very interested in doing something to funding the generation of genomic data in Brazil. And again, the level of engagement of people in Brazil demonstrate this growing interest by the scientific community to join efforts, to share knowledge, what's working, what's not working, and how can we learn together and build these collaborative strategies to address the challenge of studying and conserving neotropical biodiversity using genomic data. And also, it shows how widespread a widespread recognition that advancing genomics in the neotropics requires this coordinated action and collective investment. And following all these discussions and the symposiums and the meetings we had with several researchers in Brazil, we started what we call the Brazilian Genomics Network for Biodiversity. That is a network that unites the existing consortia that such as GBB, the infrastructure and long-term research programs and other academics from Brazil. And it aims to act as a bridge between academia, government, and society. It is structured in 3 phases over 10 years, so we are just starting it. But the idea is to develop data sharing policies, regional infrastructure, training programs, and collaborative platforms to ensure that Brazil really leads the way in biodiversity genomics at both scientific and political levels. Because the people are already doing genomics. It's not that we are starting. So we are already doing all the stuff. But this type of consortium, the type of gathering. It's so important to reach another level of discussions, speaking worldwide. Naturally, the Genotropics had to expand, as was our idea since the beginning. So now we have associated researchers that were with us in all these discussions that I mentioned, and that most of them are part of phase 2 that Camila is going to give more details. And the idea, of course, is expand even more. So the next steps. Now, to identify opportunities of external financing support. Of course, our workshop that is coming in September, and already thinking about the workshop of the next year. And mostly, Camila will talk more about this phase 2 regarding our activities. And that would be. Thank you. You want to? Yes. Yeah,
Thanks a lot, Mariana. I think if there is any question now directly, we can definitely go for it and discuss it. Otherwise, we can also leave for later. Anyone here from the audience would like to ask anything? We are following as well the chat online. So if you guys online have any questions, please post them anytime. Even if the presentation has already passed, we're going to approach your questions later on. So, but I will continue then and get started with the second presentation, where I'm going to talk about our most recent initiated project. This is a project where we aim to unveil some neotropical genomes through a network of Brazilian researchers. And this specific project then is financed by the National Research Council in Brazil. In a specific call, I will mention to you which call this is, I think in the next slide. But I think the first thing that I would like to do is to introduce our core team there. So we are two co-leaders, Mariana Teles from the Federal University of Goiás in Brazil, and also from the Catholic University, also in Goiás. She's leading from the Brazil side and is the main applicant actually from the Brazil side. And from outside of Brazil, so from the Brazilian diaspora, I'm the, let's say, international leader. And we have our team of coordination team, which is essential to guarantee the success of this project. Carmen Ruiz, who is also sitting in Goiás and will be here in Berlin very soon. Jessica Fernanda Ramos, who is here in Berlin now, and Luisa Marins, who is coordinating all of this streaming here and the making sure that our communications are done properly. So thank you all. And I'm talking here today on behalf of Mariana as well. So Mariana, if you see this now or later, thanks again a lot for taking this journey together with me, with the other three in the coordination, and also with all our partners, which include most of the Genotropics researchers, associated researchers, and core members that Mariana introduced to you, plus a number of collaborators that we have also engaged in this project. So this call was a call for 2025. So the project started earlier this year to support network projects with Brazilian researchers abroad. So it was a very special call and understanding from the Brazilian government that by activating the Brazilian scientific diaspora would be a great way to form new networks and to have even more international engagement with the research done in Brazil. So we are three researchers, Brazilian researchers who are based outside of Brazil. Apart from myself, we have Marcela Oliano, who is a researcher at the Wellcome Trust Sanger Institute in the UK, and Juliana Viana, who is a researcher at the Catholic University in Chile, and also the leader of the Chile 1000 Genomes project, which is probably the first large project in Latin America aiming at sequencing genomes from the species that we have here, or that we have in Latin America. We're not in Latin America. Also many other collaborators. Each one of them is responsible for one species, is in a little group or sub-consortium who's then responsible for the sequencing the species. So we are aiming at sequencing about 10 genomes, but this is remains to be seen because we would like to gather more genomes into this project. I will explain to you a little bit the goals and maybe even motivate some of you to reach out to us with genomes that you might be doing already, because this goes way beyond the genomes themselves, but it's a whole process of acquiring these genomes and especially making sure that researchers in Brazil can be the leaders of the genome reference genome sequencing. So this project here, funded this way by the National Research Council in Brazil, we are taking this as a pilot. So the pilot is starting in Brazil, but we have really the goal or the overarching vision that we have is to reach out to the Neotropics. So researchers sitting in the Neotropics, researchers who are somewhere else but working on neotropical species that would like to connect and work together, especially in equalitarian ways, to sequence these genomes and analyze them. So the goal of this pilot then is to strengthen genomic research in the Neotropics and promote biodiversity conservation actions by, first of all, generating these high-quality genomes for neotropical species, that each one of them fitting into a basic or applied research project. So at the end, each species here actually is going to contribute to a larger project or action. Second, and maybe on the core of this project, is developing strategies to overcome operational bottlenecks in obtaining these genomes, establishing good practices, standardizing methods, but especially overcoming the main bottlenecks and the limitations that we have in infrastructure, and in some cases also in expertise to reach the high quality genomes that we would like to. In order to do that and to have a bank of species that are going to represent a great part of maybe the reference genomes from the Neotropics, sequenced, led by neotropical based researchers, one of the aims is to start storing biological samples in scientific collections in Brazil for future studies, so that researchers in Brazil or in the Neotropics can go back to these samples in case this is needed to promote the continuity of research and maintaining ties with the country. With all of this, we also, because of there is so much research done all over the world, but more importantly, there are networks, a lot of them under the umbrella of the Earthbiogenome project, which is the EBP, which is a global network that aims to connect all of these initiatives to help them reach the quality of reference genomes from the entire eukaryotic biodiversity. So connecting to all of these initiatives in different types, in different continents, in different countries, this is going to be crucial for not only in Brazil, but in the Neotropics to reach the capacity of sequencing these genomes. In this specific project, the participation of Brazilian researchers abroad was very important and will remain very important. I think Genotropics, one of the maybe strengths of it is already, and is going to be more and more to activate this diaspora. So all the researchers from Latin America, that are sitting somewhere else that already work with genomics that would like to engage with their countries of origin and support this process. So, okay, I'm talking about all these challenges of obtaining reference genomes. And in order to understand what these challenges could be, we need to look at the workflow of assembling genomes. So it starts even before one collects samples to obtain the licenses that are needed. These licenses, it's not only collecting licenses, but nowadays there is much more. We have, as we are going to put data openly available in public repositories, we have to think about the fact that this data is going to be made available internationally. So there is the program, the Nagoya Protocol, which is the basis of what is regulating this access to this type of data. And Brazil, for example, has very specific ways of controlling this process. I think probably it's one of the countries that has the most developed system. Does not mean it's an easy system, but it's quite a well-developed system with specific goals. So it's easy compared to other countries nowadays to actually register the species that are going to be sequenced, that are going to be maybe shipped abroad, but also for which the data is going to be made available in public repositories around the world. We cannot control anymore. No, the genetic resources, they are not just the physical ones anymore, they are the digital ones as well. And this is what is being taken into consideration here. Second, the collection and shipment of samples. For sequencing reference genomes, we need samples, we still need samples that are very high quality, that ideally have DNA that is preserved and also cells, including the nuclei, that are preserved so that we can reconstruct properly this structure of the genomes using different types of technologies. There is, I would say, approaches that are being developed in order to try to break this cold chain, in order to make it easier to have these collections done in remote areas and less dependent on liquid nitrogen. But it is true that's the best way to guarantee a high quality DNA. You should do it with liquid nitrogen, super fresh samples. That would be the ideal procedure. Okay, so this brings some limitations, of course. The sequencing itself takes different technologies that are not always accessible for universities and institutes in different countries. So mostly long reads. I think PacBio hi-fi reads are the maybe state of the art. Nanopore, Oxford Nanopore sequencing is getting more and more popular. It's more experimental, let's say, than PacBio, but also quite potentially very powerful technology that I believe, especially in countries like Brazil and other Latin American ones, is probably going to be the main technology for most of the labs who are generating data inside the country. Then we have the Hi-C, which is this chromatin conformational analysis where we try to get the chromatin folding structure inside the nuclei to help us reconstruct the chromosomes and link and build further from the first scaffolds or contigs, that we managed to assemble after we have done the first sequencing. The Hi-C, similarly, for now, is state of the art. It does not mean that it's always needed, but it's highly desirable. And for many, many species, it is still needed. So, but there is development there. Hopefully, things will be made easier. And just for you to know, nowadays, almost as far as we know, almost no one in Brazil can do the Hi-C for this type of sequencing. I think apart from the GBB, so from the Valley Technology Institute, we are not aware of others that are importing kits and building, even though this is a technology that many labs from the past doing cytogenetics and other types of analysis of chromatin actually have the technology, but they're not necessarily using nowadays for assembling genomes. And then we go to the computer, And that's where the expertise in assembling the genomes and maybe more importantly, curating the assembly of the genomes take place. So in every one of these steps, there will be limitations depending on how these researchers are going to sequence the genomes. And here we have thought of 4 different logistic models that we are testing inside this project. The first model includes the infrastructure in Brazil, so where all the data needs to be created, produced in Brazil, and the genomes are then assembled also in Brazil. The second one is via international collaboration, and this is, of course, something that many already do, so they ship their samples abroad. But the idea here is to make this, well, it's not only a shipment of samples, it's a participation in the process by at least having a local biobank with a voucher of this sample that are going to be sequenced representing this species in the public repositories. Also, we need to streamline the documentation, making it sure that there is the documentation within the country that this sample is there and is being used for this purpose, in order to, if needed, think about benefit sharing. So I think this is one of the aspects that also Genotropics will be more and more involved when thinking about international collaborations, because the benefit sharing, so meaning the fact that whoever is accessing this data, this information abroad, so not in the country of origin, should think that for this to be there, this came from biodiversity sitting somewhere. And in many, many cases, this place does not have the same capabilities or resources to analyze the data of their own biodiversity. So by having the privilege of accessing this information, the sharing of benefits really have to be thought over. And I think this is going to be a big topic for the next years, especially when we think about the Cali Fund from the last COP of the Convention on Biological Diversity, which is something that is going to be defined in the coming years. Then we have the Model 3, again, something that Brazilian researchers or Brazil-situated researchers use commonly, companies abroad. So they ship their samples. The data is theirs. Yeah? So it's my data, I'm shipping, I'm paying for a service, but there are problems there. Still, documentation sometimes is problematic. Second, the cold chain, which is always part of it. And last but not least, the hybrid mode, where part of this data might be created in Brazil and part of the data still needs to be created abroad because maybe there is no access to the technology in Brazil, or maybe it's too expensive. So we wanted to be as versatile as possible in thinking about these different models, because we believe that each researcher will more or less pick one of them in their normal life. And we should think about the bottlenecks that each one of them present nowadays, so that we try to mitigate these bottlenecks and hopefully make the groups sitting in the Neotropics able to generate their genome researchers in the mode that they want. Right? It doesn't matter what it is, but following certain requirements, certain ethical principles, and so on. So here we have another way of representing this model, where the bottlenecks. So where you can see a little bit more where the most important bottlenecks are for each of the different models. So part of them, the infrastructure in Brazil, we still have a trouble with sequencing because of access to certain technologies. Even though Oxford Nanopore, as I said, is increasing theirs, but there is a big learning curve to be, to be traveled through. And High-C apparently is still a big bottleneck and something that we should think about. When shipping things abroad, whether by international collaboration and companies, the licensing part is a very important point, and the cold chain, and also the biobanking locally. And the hybrid strategy, then it really depends on what the choices are. So we want to approach many of these bottlenecks thinking beyond just the technology, the availability, but also what are the costs, because the costs will, of course, make or guide the researchers into choosing one of these models. What is the final genome quality that each of these models can present? So if you have some limitation on what you can ship, so many, for example, are deciding to ship DNA because it's just easier. If you have to ship sample like tissue, you're going to have way more paperwork to do. And in many cases, it's going to go back to your institute because you have to go through customs, through other types of sanitary controls and so on. So DNA is still easier, but then there are limitations if you're just shipping DNA abroad. No, there might be limitations on what can be done with this DNA. If it's not long enough, high quality enough, if for high C, for example, this is not possible to be done just by extracting DNA, and so on and so forth. So there's limitations there depending on the model that is of choice. Also, how easy it is to follow all these steps of each model and how we can circumvent the bottlenecks. So the way that we are doing this is, of course, by imagining every one of these steps, using our own experience. But more importantly, we are doing this by in practice, like it's the learning by doing, and having all these researchers, for now, 10 of them, that are distributed, they're in different institutions with different levels of expertise and maybe resources in their own institutions. And each one of them trying to get through one of these models, and us from the coordination trying to guide this process and document and see where we can circumvent some of the bottlenecks that appear. Here we have the representation of 8 of the possible species. So that if they are going to be sequenced remains to be seen, because we are right in the process of defining, making sure that samples can be collected along with the permits that are needed, and so on and so forth. But we have plants, we have mammals, birds, insects, fish. So we try to be diverse, but also respecting the interests of the researchers that wanted to have the genomes for their own projects. And some of them have already started, and many of them are still in the planning phase. So I'm hoping that by early next year, we're going to have some results to show and share with everyone. Yes, I think that's it from me. And thank you, everyone. Do you have any questions here in the locally? Yes. Do we have a microphone or? Okay, just say it and I repeat.
I was wondering, in the slide that you have with the models,
yes,
if you have any idea of the timeline, because that slide explains the bottlenecks in Brazil versus outside. And I was just wondering if some of these models require much longer time than others right now.
So the question is if one of these models requires much more time than the other. To be fair, we don't know yet, because each species is a different case, and it's really dependent on the researcher. However, what we can say is that the accessing to the sample, it will really depend. For example, for a mammal, it can be much more problematic because you have different types of permits than if you need to collect an insect for maybe which is in your backyard and so on, or for which already there is an expedition planned. So we are seeing huge differences in the timing for that, depending on the species and the researchers who are trying to do that. So this is already something, though, that it depends. But let's say if the samples would be ready to go, then I'm doing a guessing work now. I would say that most likely the companies abroad would be the quickest, just because usually commercially they will give you a time. It's going to cost maybe more than doing with a collaborator and so on. But they will give you some time frame where they say, well, you're going to get the data back within this time frame. And then it will depend on what? If the company actually can deliver what they promised. And this is the other thing that we are also testing. So we are testing a couple of companies, mostly in the US. Now we don't know what's going to happen in the US, but maybe one in Canada and maybe one in Korea, for which, the researchers are already using. So we are not reinventing the wheel. We're just getting whatever the Brazilian researchers are already using and seeing if we can interact with them, make deals, see how much information do they give us back. Because a lot of these companies, they might give you the data, they're not going to tell you exactly what they did, which is also, not ideal. So there are disadvantages, but I would say, in principle, if they get the shipment, they would be the fastest, for sure. The infrastructure in Brazil, we are now starting to work really hard in getting the labs that have oxynanopore technology with the Promethean 2, so P2. There are two labs within the project, and already now a third lab that we are connecting to with a lot of experience from Paraná. We have partnered them with labs in Chile. So I mentioned the 1,000 Chilean genomes. They are already way ahead in the sense that they are producing long reads from oxynanopore for many different species, endemic species, plants and vertebrates, invertebrates, and so on in Chile. And we had a meeting where we joined 4 labs in Brazil, 4 labs from Chile, and they were exchanging their experiences. So this is the type of things that we're trying to promote, to see if we can actually consider that the oxynanopore will be a, let's say, successful model for the model number 1. I mean, if the sequencing is working and is enough, the model number 1 will be the fastest, but it depends on us and our collaborators and the others being able to overcome the bottlenecks. That's basically, so for now, we cannot do the timing. We will do at the end of the project, so we can tell you back which one took the longest and so on. Yes, do you want to compliment?
Yeah,
but then come over here, please.
I'll just say that I think it's a great idea to track
but we will do this, yeah. We'll track it.
The quality, the time of each step is a good idea that you track.
Yeah.
And
we're definitely going to track costs, the timing, the whole procedure, to also see what could we have done, to make the whole thing easier and smoother and so on. So it's really a learning by doing process that we are going through, but otherwise it's all too theoretical. And one thing that I think we have learned, or I at least have learned from working at the European level, is that you cannot just transfer your experience from one side to one country to the other without knowing the exact conditions that you're going to find in these other countries. So then it's this type of experience we are going through. Yes, and you have a question?
Because we consider the RNA and the logistics planning.
Yeah, that's a really good point. Yes, we want to. Oh yeah, transcriptomes. Do we intend to sequence the transcriptomes? Yes, we intend to sequence the transcriptomes to finalize the genomes and the annotation of the genomes. But for now, we don't consider that this is the major challenge, because this is Illumina sequencing based. I mean, maybe if you would be thinking about ISO-seq, the PacBio version, it would be a challenge, but it's Illumina sequencing based, and many labs are already doing this, I would say most likely internationally. So they send abroad for sequencing, but it's definitely in the plans. It's just not the priority, because first we need to make it sure that we can assemble. Yeah. In the sample collection. Yes. Yes, we are motivating all the groups, the PIs and their groups, to collect samples that are going to be useful for transcriptomics. But of course, we are only going to do it once we know that we're going to have a genome. Yeah, and I also I don't know for each case if they're going to manage at first. No, it might be that this is going to be a second round where they're going to go and search for the tissues and so on, because this is also, from our experience, I can give you an experience here from ITSW. So we sequenced a couple of genomes from the samples that came from the pathology collection here. And usually you use one tissue for sequencing the genome, so to get the DNA and the nuclei and so on. And that's usually what we did first. And then after that, I mean, ideally you do this in parallel, but then after that, we went on and got all the tissues that would be good enough for transcriptomics. And in those cases where we didn't have them, then we had to maybe wait. So we had a big project with sea turtles. And in some cases, the researchers went there, they collected blood from living sea turtles, and they had to wait until there was a stranded sea turtle. And in some cases, it happened a year after that, which is fine, because we are talking about having a legacy here, about building a database of reference genomes that are going to be high quality. So even we don't have to rush into doing things, but ideally, if we can sample at the same time, that would be the ideal. Eduardo, do you want to comment? Or
I was just going to comment on the transcriptome, but maybe I will when I'm there.
Yeah, or you come over here right now.
Questions?
There are questions there. Okay. Well, could you tell me, because what is the first one?
Biobank plan for storing biological samples, or will each researcher be responsible for storing that sample?
Great question. And if I had time to prepare this a little bit more, I would have put all the details. So thank you for the question. What do we have a central biobank? Yes, we are piloting, and this is an important point, because so there is a biobank, and the Oswaldo Cruz Foundation, which is an institution that is, let's say, funded by the health ministry in Brazil. And they have a very large biobank that they use for COVID. And now they are transforming this biobank into health and biodiversity. And they are partners in this project. And we are going to, let's say, pilot the process of biobanking there. But we have to be aware that this biobanking is it's complicated, it's bureaucratical. So this whole business has to be developed, which means I don't want to promise anything that by the end of it, we'll have the entire process figured out and done. But that's the goal, okay? That will be a specific biobank for this pilot, which does not mean that in the future there won't be other biobanks sitting in Brazil. So I think this is, if this project gets larger, if this network gets larger and so on, ideally there should be different biobanks distributed across the Neotropics, Brazil being big, most likely. It's going to have more than one biobank, but it remains to be seen. So, yeah. Anything else?
Yes.
Do you want to come over here maybe, and you can say in the microphone?
Rodrigo is asking, is there any strategy to join the whole world in a single effort, Europe, Brazil, China, and the US?
Well, there is the Earthbiogenome project, which is a network of networks. So it's not a network in the sense there isn't one big plan for the globe. Also because, can you imagine one big plan for the globe working? Probably not. But, networks, projects that get under the same umbrella, that agree on certain principles, I think that's what the Earthbiogenome project is trying to do. Which does not mean, for example, that networks cannot partner with others. So we have, I'm part from the European Reference Genome Atlas in Europe. And one of the goals is also partner with other networks directly, because the Earthbiogenome project is this umbrella initiative. It's not the hands-on. So sometimes the problems, the everyday type of problems, you can only see and share with others when you connect networks directly to each other. But the most important thing is that they are become under the same umbrella, accepting certain principles of open science, of ethics, benefit sharing, and so on. I think this is, and quality. So I think we all want to reach the quality for genomes, to have this as I said, as a legacy. So reaching the state of the art. Now we want the world to be able to use the same state of the art technology that is available. So I think that's part of also the Earthbiogenome project, I would say, philosophy, and something that we also try in Genotropics to support, so that this is possible in the Neotropics.
Next one, Enzo. Hi, Enzo Segnotti from Instituto Missionario de Biodiversidade, Argentina. First of all, congratulations on your presentation. Very clear, interesting. My question has to do with database administration. What softwares do you store, manage, track samples, specimens, and metadata?
Software, Google Sheets. We at this project, that's a small project. I'm super thankful for that, because in the very large project in Europe, we still struggle to have something that is functional, which is central and everybody uses in the same way. Because once you distribute, it's easy when it's inside one institute. Okay, it's a big institute, it's sequencing a lot. There you can use the same database, it's gonna work probably like a charm. But from the moment that you have different centers generating data, having their own workflows, pipelines, and then trying to centralize this, it gets complicated. So we are starting, this is not what we are facing right now in this project, not this specific project, but I mean, it will come, and we're gonna have to, if this gets bigger, we're gonna have to take decisions whether this database, we need to have a database to track everything, and so on and so forth. So to some level, yes. I don't think there is a way for me to share anything now, but for link and so on. But in the ERGA, there is a portal which is done by the European Bioinformatics Institute, the EBI. And this portal, what it basically gathers information from public repositories. So after the whole process of us uploading things to the public repositories, they gather the information, the metadata, and so on, and they present this, let's say, to the world. So that's a nice way to present to the public. But an internal tracking, we have tried different things. It is tricky to do this at a large scale. And yeah, we don't have a proper plan now for this network, but happy to discuss if people have ideas and would like to reach out.
Just the last question.
The last one? Yeah.
Pedro is asking, once you have the expertise, what is the main bottleneck in nanopore technology today for reference genomes?
Once you have the expertise, okay. If by expertise, meaning if you know how to sequence and you can handle the extraction, the library prep, and the sequencing itself, I would say is having samples that will give you enough DNA, because Oxford Nanopore needs more DNA than PacBio, for example. For PacBio, you even have this ultra low input option. So for Oxford Nanopore, you do need more. So if you're dealing with tiny, tiny stuff, it might be tricky depending on the, how this DNA is extracted and the amounts of it. But I don't know what's going to happen in the future. However, let's say if this is not the problem, and if you have a general expertise, I think one problem with Oxford Nanopore that I hear from different centers is that usually it doesn't behave the same way, even if you're doing for the same thing twice. So it's a technology that demands a lot of dedication. And understanding and might be very different depending on what you're handling. If it's plants, if it's vertebrates, if it's especially then invertebrates with loads of symbionts and other stuff. So or secondary metabolites, and so I would say it's less standardizable. than PacBio, but I can see that the ones that get the hang of it, they usually can always or almost always get good data, especially if it's coming from the same type of taxa. But the diversity of taxa creates challenges for a lab running Nanopore. Anything else for now? No. Okay. Good. Here. Sorry, in person. Do you guys have questions? No. Then thank you so much. I will now pass on the words to Eduardo, who is going to continue presenting a new institute that is going to start in Brazil very soon.
All right. So I hope people hear me all right. Can you check, Luisa? Am I showing on the screen and people are hearing me? Please. So thanks, Mari and Camila. This has been great. I'm excited to present for the first time this institute that got funded 10 days ago. So this is the first presentation on this institute. It's going to be called the Brazilian Institute of Biodiversity Genomics. In Portuguese is INCT, which is called the National Institute of Science and Technology, which are many. I'll show in the next slide, in biodiversity genomics. To change the slides is here, or this one. So as I was introduced earlier, my name is Eduardo. I'm a professor in Southern Brazil at this Pontifical Catholic University. I lead a research group mostly focused on carnivores and evolutionary and conservation genomics. This is the Laboratory of Genomics and Molecular Biology. I'm also a member of this NGO, the Procarnivorous Institute. That's an NGO focused on carnivore conservation. And I'm also a member of Genotropics, like Mari and Camila presented earlier. And this institute that we just got funded is funded by CNPq, our National Research Council, the same council that is funding our biogenomes project. So I'm a member of Genotropics and the new project that Camila talked about. And now we have this newer project funded. So this is a screenshot from the MCTI, the Brazilian Ministry of Science and Technology website, showing that the INCT is one of the largest programs in science and technology in Brazil, probably the largest. They've been funding billions of dollars worth of science over the last 12 years or 15 years or so. They've had different calls, like in 2010, 2014, 2022. There were about 200 institutes across all areas of science funded in Brazil. And in this recent call, 2024, 143 institutes are funded across all areas of science, technology, engineering, humanities. And that includes some renewal. So actually, we don't know yet how many of these 143 are new ones. Ours is a new one. And some of them will probably be renewals of the 200 that were already funded before. And surprisingly, or happily for us, ours was ranked first among all 143 that were funded this year. So it's quite amazing to see how enthusiastic the assessors were, the evaluators were about biodiversity genomics. So this is the first national institute in Brazil devoted to biodiversity genomics. So it's really, really rewarding to see this, and I'm excited to show this and to get started. These are the institutions that are involved. So this is a large project. It's a network-based project. We have 17 Brazilian institutions involved. This is a list. I'm not going to read all the list. Several universities. So the universities are in blue. The headquarters will be here in Southern Brazil, which is where I work, and other colleagues work as well. The vice chair is Fernanda Werneck from Amazonas, from the National Institute of Amazonian Research here in Manaus. And then we have universities from all these areas in Brazil. And then the yellow dots are technology institutes. So both public and private, a non-profit private university center institute, which is what Mari and Camila also mentioned, ITV. They're also members of this INCT. This is the same map, but now showing the people. So the steering committee are the two of us, the chair and vice chair, but also Mari, who's here, Cristina Miyake, Fred Henning, and Sandro Bonato. So Chris Miyake is from USP, from the University of São Paulo. Fred is from the Federal University of Rio de Janeiro in Rio. Mari is from Unicamp, and Sandro is also from PUCRS, from my university. So we are the steering committee. These are the PIs based in Brazil. So I'm not going to read all the names. We can go back later about what they do, what areas they work on. But we have 30 Brazilian PIs working at the institute. Each of them leads a group. And then we have 4 international partners who are part of the proposal, who are here. So Camila and Ricardo are here, and Juliana Viana and Marcelo Liano Silva, who were already mentioned, are also partners in Genotropics. And in this project that Camila just talked about, they're also partners at the INCT. So the funding we got so far, and probably they're going to supplement it over the years, but for now it's a 5-year project, funded 12 million reais. It's the currency in Brazil, which is about equivalent to $2.2 million, to do a bunch of different things. So I just listed, this is going to be like a short presentation on the Institute, and then I added some slides that are examples of things we're already doing that maybe will be interesting for the audience. If not, I'll just stop and just talk to you guys about the prospects. But these are the research axes. There are 15 axes in the Institute. That's what we propose, right? We haven't even had, you guys are hearing about it before we even have a meeting of all the PIs, right? We haven't had the meeting of all the PIs yet. We're trying to schedule one, but maybe some of them will be seeing us here. And this is what they already know, because all I'm talking about is what was in the proposal. So that's what we want to do. Axis 1 is reference genomes. So it follows up on what Camila just presented. So basically the current project that's also funded by CNPq will be the seed for the Axis 1 here. So basically it's going to be to continue and expand on what Camila just presented, and then to try to include other taxa, basically explore poorly known taxa, for example, single-celled eukaryotes, small metazoans that are poorly known, taxa that have been neglected by other projects internationally, trying to contribute new orders, new families, new genera that have not been sequenced by other EBP-associated projects, in addition to taxa that are the focus of research already, like the ones that Marian and Camila presented. Then Axis 2 is phylogenomics and evolutionary genomics. So basically figuring out phylogenetic relationships, hybridization, adaptive divergence. So a lot of the research we are already conducting. So of course, these don't depend on the reference genomes, but they can be greatly improved by using high quality reference genomes. So a lot of the research we have already been doing in my group and other colleagues' groups relate to phylogenomics and evolutionary genomics. Mariana's research as well focused on this axis. So myself, Mariana, and several other colleagues work mostly on Axis 2. I also work on Axis 3, which I'm just going to talk about briefly. So this one will include a lot of the deeper level phylogenomic projects that we are already doing and want to improve on. Axis 3, population conservation genomics, mostly using resequencing, so Illumina data. We're distinguishing different kinds of population genomics. So Axis 3 is what we're already doing as well. So basically resequencing at the population level, using Illumina technology to look at population structure, genomic diversity, runs of homozygosity, load, various kinds of problems that we are already studying in different groups. So expanding on that, connecting people, improving analyses, exchanging. So the INCT, the Institute, is about exchanging and improving capabilities. So that's going to be mentioned in the next slide. But it's basically leveraging what we've already doing at Genotropics and what Camila just presented in the second phase of Genotropics. So expanding the connections, expanding, knowledge transfer, understanding the different approaches and how they can solve problems. The fourth is the field that is mostly the focus of Fernanda Vernex and her colleagues, which is studying ecogenomics. So also population level analysis, also mostly using Illumina, at least for now, but mostly looking at functional variants and functional traits related to genomic diversity, especially in what concerns predictions of climate change impacts. So predicting and projecting into the future genomic diversity, both neutral and adaptive, and how it affects, vulnerability at the genomic level, cases of rescue that can be promoted in terms of reconnecting populations. So mostly related to functional adaptive diversity in the face of climate change. Then the fifth axis is also population genomics, also mostly using Illumina. So still current technology. And of course, all of them can take advantage of reference genomes. So reference genomes can improve on all of them. But here we focus on species of interest to the bioeconomy. We have a single species selected so far in the proposal, which is the yerba mate. So because we're based in southern Brazil, we thought it was kind of a nice choice. And we already have a group working on yerba mate improvement, genetic improvement, and they don't have a lot of genomic data yet. So they joined in. We have 3 people from the yerba mate project that joined our proposal. So now they're really excited to soon have genomic data. Basically, we're going to sequence a lot of yerba mate strains and provide them with SNP data to construct a chip for improved breeding. improved breeding and genetic management of yerba mate strains for improved production. So I think that's a very clear application of genomic data for the bioeconomy that will be really nice for southern Brazil. I mean, you know, southern Brazil is really into yerba mate, and Argentina and Paraguay as well. So I think it's going to be a very popular project. We can probably make a lot of publicity with the yerba mate one. But yeah, so that's part of what we want to do. Then there's axis 6, which is pangenomics. So here we're talking about population genomics with long read data. So building high quality genomes at the population level. So this is the field that Fred Henning is our main expert in the project. And so he's already doing that with his cichlid system. He's already part of that other project that Camila just mentioned. That's one of the reference genomes we're going to build in that other project. So here we're going to take that to another level. He's already doing that, but we're going to hopefully expand in numbers and also start at least a second system during the time of this project. So we haven't chosen the second system yet. So it's going to be one of Fred's systems plus a second system. So during the project, we're going to choose another one and explore that. 7th is museomics. That's an axis that Ricardo for sure will be very involved in. We also have Joyce Prada and Gabriela Camacho, who are the Zoological Museum in USP, in Universidade de São Paulo. They're setting up a museomics lab there, and we have other colleagues in Brazil interested in museomics. We have already been doing museomics ourselves, but in collaboration with people in other countries, mostly US and Europe. So the idea is actually to be able to generate museomics data that is generating genome level data from museum specimens, mostly Illumina for now. I mean, I don't think there's any prospect of long read data, because DNA is very degraded. So mostly Illumina from museum specimens to address questions that are likely to be connected to axis 2 and 3. So it's not like you need a question only for the museomics axis. This is mostly a methodological axis related in terms of scientific questions to axis 2 and 3. So phylogenomic, phylogeographic, population genomic questions. Then you have integration axis up to 15. So there was a question about biobanks that was just asked. So the axis 8 has to do with what Camila just answered. So the goal of axis 8 is to leverage what has already been planned and is already in the works in this project that Camila just presented, and then to integrate it some more. So Manuela da Silva at Fiocruz is also part of INCT. So the idea is to not only work with the Fiocruz biobank, but to promote the origin and improvement of biobanks in multiple sites in Brazil. So we can fund some small-scale biobanks, so promoting good practices in terms of metadata, how to connect the biobanks, how to have redundancy. So for example, having parts of the samples in one and then replicates in other biobanks, connecting the collections. We have people in the project among the PIs that are curators in scientific collections. So how to connect. Some scientific collections only have, for example, talking about mammals, my own field, they have skulls and pelts. So they will be related, for example, with the museomics axis. But some of them collect specimens, and so they collect tissue. So they have tissue in ethanol, or they have tissue in freezers. That could be a biobank, and they already have some that are organized biobanks. Other people are starting to do fibroblast, viably frozen fibroblast cell cultures. That's a much better quality source of DNA that will be used for a reference genome. So the idea of axis 8 is to integrate scientific collections in its various shapes and forms with biobanks. Sometimes the collection and the biobank will be the same. Sometimes they'll be connected. Several institutions in Brazil have one or the other, or are starting one and have the other. So the idea is to map what we have, connect what we have, and improve on what we have. So that's axis 8. Axis 9 is capacity building, training, protocol. There's an L missing there. Standardization, best practices. So again, building up on what Camila just presented. So improving, capacity building, training, and Mariana also presented that Genotropics has a very strong training component. So this will be expanding on what Genotropics is already doing and what GBB is already doing. They already have training workshops as well. So the idea is to connect and promote synergies. And the course we're teaching in September, the population genomics course, is a Genotropics event, but it will already help start some of the INCT initiatives. But the idea is not to reinvent the wheel and not to duplicate efforts, but rather to connect and complement what we're already doing. Expand the international scientific collaborations, people have already mentioned, so that's an axis, but we're already doing that. Increase Brazilian participation in international genomics consortia, especially EBP, but others. Our project has been endorsed by EBP, by IRGA, by the Threatened Species Initiative in Australia. as well as by the Darwin Tree of Life initiative in the UK. So we already have good ties with all these consortium that is to promote, again, along the lines that was the question that was just asked about the worldwide interactions. We're trying to help as much as we can in connections to the global initiatives. 12, expansion and transfer of technology and genomic knowledge to private and public sector. So this is more of the applied component. This was important as part of the proposal. So basically enabling using genome data, both public policy and technological economic development. Then the 13th is genomic enablement of NGOs working on biodiversity conservation. So 12 and 13 are more of how we're going to transfer genomic knowledge, so improving our connection. A lot of our PIs already work with government agencies, with NGOs, and with the private sector. So how can we improve on that, and how can we improve our efficiency in transferring knowledge and using that for various types of applications? In 14 and 15 are communication strategies. The first is to publicize the scientific discoveries and their impact, that we derive from the study, in the various studies that we conduct. And the 15th is popularizing genomic sciences in general and sensitizing people to how important they are and how they can help scientific development in general. So these are the various X's. This is what we hope to do. And what I would have now are examples. So because that's where we are. We just got funded 10 days ago. So I don't know if you want to ask questions about the INCT, or if I should go into some examples of studies in our group that basically I picked studies from 2 and 3, phylogenomics and evolutionary genomics. This is not the main focus of the symposium, which was basically to showcase what Genotropics is doing and our current and future initiatives, but if people are interested, we could do that. They are. There's one person at least. Yeah. One is enough. Two are enough. Well, Ricardo has seen a lot of them. So, okay, just a few slides. So just on axis 2, phylogenomics, evolutionary genomics from my own group, things we've already been doing. And it includes museomics in this case. So that's axis 7. So in this case, this is a paper that got out a few years ago, 3 years ago, on otters. This was a PhD paper from Vera de Ferran in my group. She was co-advised by Klaus Kipfli, who's at the Smithsonian Mason School of Conservation in the US. These are the authors. Several of them are people who recurrently collaborate in projects. We have, for example, Juliana Viana, who's our Chilean collaborator. She got this picture. This is a very endangered Chilean otter. She's the one that sampled this otter and got the picture of the cover. So Juliana is leading the 1,000 Genomes project in Chile, collaborating with Cristobal, who's here. And so she was an important partner in this study. Here we collected 14 novel genomes from all the species of otters in the world. Actually, I've just brought one slide per study, so I'm not going to show the graphs. I'm just going to tell you the stories of the different papers, and if you're interested, I can tell you more details. So we sequenced all the otter species in the world up to then, which was 13 species. Now we found the 14th species, but we waited to publish the 14th species before we published this one, so we could say we sequenced all the 13 otter species in the world. 14 of them were novel individuals, and 2 of them were from museum specimens. So museum specimens held in Paris and Copenhagen. So that was our first experience with museomics. So we did that with partners in Europe, in Copenhagen, Tom Gilbert's lab. So we got those sequences from an African species and a Southeast Asian species, which was important for us to test some of our hypotheses about species diversification in otters. We also looked at diversity in different species. I'll have one slide later on comparing diversity for this otter, that's very depleted in diversity in southern Chile. But that was an example of a phylogenomics and evolutionary genomics study. This other paper also came out in 2022. This was led by Jorge Ramirez. He's a Peruvian professor who was at the time a postdoc in Pedro Galetti's lab. So Pedro is also a partner in Genotropics, also a partner in this new project that Camila talked about. Patricia Freitas is also a partner in Genotropics and the new study. So I'm just showcasing studies that include collaborators just to show that the network is already working, right? Basically, that's to show that. In this paper, we sequenced for the first time a genome of the margay, which is a very arboreal cat in South America, the most arboreal species we have there. I mostly focus on cats, most of my research, but I do other carnivores as well. And then we did a wild-caught ocelot. So we found, and I'm going to have a slide in a few minutes, showing that the ocelot is the most diverse cat in the world. So the heterozygosity genome-wide of ocelots is higher than any other cat. And it's the sister species, or at least we thought so, the sister species of the margay. Now we found another species that's closer to the margay, but we looked at not only the phylogeny, but mostly adaptive divergence. So signatures of positive selection in these species. We found an interesting gene that's involved in retinal development that has a signature of positive selection in the margay. The margay has huge eyes, the largest eyes among the Neotropical cats. And this gene involved in retinal development has a signature of positive selection in the margay. And that was one of our main spins of this paper. This was another paper now on canids. So this is another family of carnivores that we work on. This was led by Daniel Chavez, who's an Ecuadorian professor now. He did his PhD with Bob Wayne at the University of California. He took a course with us in 2011 as an undergrad, and then went on to do a PhD with Bob. He met Bob at the course we organized. That just shows how powerful and productive these courses can be. So we organized this course in Brazil. It's the ConGen course, the Conservation Genetics course that we have every year or two globally. So it's called ConGen Global, conservationgenetics.org. So putting a plug for our ConGen. He took our ConGen edition in 2011 in Brazil. It's the only edition in South America so far. And there he met Bob, where Bob was one of the PIs, one of the professors. Unfortunately, Bob is deceased now, but at the time, he was one of the founders and leaders in canid genomics research. And so he went to do his PhD with Bob, and Klaus and I helped direct the study. The original idea was to compare the maned wolf and the bush dog. These are sister species that are remarkably different. This one is an omnivorous species. There's a wolf fruit here. Some areas of its range, it eats half of its diet is this fruit. And this is a hypercarnivorous form, only eats meat. This is social, this is solitary, this is a forest dwelling, although it also lives in some cerrado and some savanna habitat. This is more of a grassland savanna. This has long legs, this has short legs. So they're very disparate. So the goal was to look at adaptive divergence. So we found some interesting genes and interesting non-coding regions that are associated with pathways related to fruit digestion and also limb elongation in these species. And we ended up sequencing the genomes from all the South American canids and comparing their history, their diversity, looking at dates, looking at biogeography. So it was a pretty comprehensive study that we continue to follow up on in various ways. For example, Cristobal, who's here, is now following up on one of the species that we studied under Daniel's supervision here at the IZW. So looking at the fox in South America, in the future, in the near future, we'll hear more about their discoveries in one of the foxes. And I'm gonna, just mention that now we just got in another project started on hologenomics of the maned wolf. So looking at whole genomes across the range, epigenomics and microbiomes across the range. So the first hologenomic study that we pursue will be on the maned wolf. Another one that came out this year was Sarah Santos's master's project. It was a phylogenomic study on jaguar, lion, and leopard. It was a very interesting collaboration with Bill Murphy's lab, a longtime friend and collaborator, but also Ziheng Yang's lab with his postdoc Thomas Fleury, who are very skilled at developing models for understanding species diversification under the multi-species coalescent model, but also including introgression. So this is a use of the MSCI model. And what we found was that lion and leopard massively hybridized in the past, which is a pretty remarkable discovery because this has been a puzzle for decades. We've been doing cat phylogenetics for 20 years, first using small pieces of DNA, then whole genomes, and still they've been hard to solve. The relationships among the Panthera cats have been hard to solve. And maybe I'll move this to the side. And basically, there's no tree that's right. The tree changes across the genome. When we found that when we sequenced the jaguar genome, that was the first genome we sequenced in Brazil, the jaguar genome in 2014. And we see that the tree changes across the genome, and sometimes the lion goes with, most of the time the lion goes with the leopard, but sometimes it goes with the jaguar. Sometimes leopard and jaguar go together. And it's basically an intractable problem until you take into account hybridization. And so the bottom line in this study is that we infer that there has been first a divergence between this lineage, the green lineage here, and then the jaguar and lion, well, at least the jaguar and the brown lineage split. And then there has been massive hybridization here. And then this massive hybridization led to lion and leopard. And if you assume this tree with jaguar and lion or this tree with jaguar and leopard, you get the same result. So basically these two trees are indistinguishable. They are undefinable statistically. They fit the data equally well. And the tree that dominates the genome is neither. The genome is dominated by lion leopard going together. But we find out that this is actually due to massive hybridization, that the signal that led to the emergence of the current species is completely obliterated by massive hybridization between lion and leopard. So it's a pretty intriguing study. Examples of population genomics. This is from Jonas Leskroart's PhD. He's a Belgian PhD student that I'm co-advising with Hannes Svardal at the University of Antwerp. This is just to show diversity, heterozygosity measures among cats. These are the ocelots that I mentioned earlier that have the highest diversity in cats. These are from South America. These are other cats from, well, actually in this paper, all of them are from South America. We had another paper with cats from other regions. And I'm just showing this one, this guy, the Guinea from southern Chile with very low diversity. And comparing this to the fox that Cristobal studies and to that otter that's on the cover, there emerged this pattern, which is quite interesting. So the pattern is that these are species from 3 different families of carnivores. All of them are endemic to this region of Chile and a little bit of Argentina here. And they all have very low diversity. They have long runs of homozygosity. They have PSMC patterns, which are patterns of historical demography that indicate a history of loss of diversity, loss of range due to glaciation, perhaps also compounded by human habitat change. So this is an interesting backdrop against which now people can do in-depth conservation genomic studies. And our main species is jaguars. So this is a study that we did on jaguars showing runs of homozygosity with species individuals from the Atlantic forest showing long runs of homozygosity. This is the sum of the length of runs of homozygosity in the genome and the number of runs. Have an individual from Arizona here with lots of small runs of homozygosity and individuals from the Atlantic forest with very long runs indicating inbreeding. And this is a focus of study that we've been pursuing, along with the use of genomic markers for applied conservation genetics. So this paper just came out as well from Gabriele Lazzari's master's degree. We use the genome to develop a SNP panel to track the origin of jaguars for confiscated jaguar parts, but also to look at individual ID, parentage, melanism detection, and other molecular ecology questions. So the hope is that we're going to produce a public database that's available for all the range countries, that's going to be fed by the studies as we generate more data on paternity and on different types of molecular ecology questions. We're going to help the forensic analysis to pinpoint the origin of jaguar parts to help combat traffic of jaguar parts. So that's published in Ecology and Evolution, and it illustrates a vision that I suggested a few months ago of how I think conservation genomics can work, and it's already working in several places. And I think it's going to drive a lot of our Axis 3 in the INCT at the Institute. So hopefully we're going to apply that to more systems. So basically using high quality reference genomes along with transcriptomes. So that reminds me of the transcriptome question. So in the jaguar genome, when we did it in 2014, we collected 6 tissues at the same time. So we got 6 tissues, and that was useful for annotation. And now it's going to be useful again, because we now have a new reference genome generated by ITV, and we're going to use the same transcriptomes that we generated a few years ago. And hopefully for the coati, for the otter, and as many systems as possible, we're going to try to do the same thing. But as Camila said, it's tricky because if we get an animal in the field, it's hard to collect many tissues. We generally get only blood and a skin biopsy for fibroblast cell culture. But we're trying to get zoo animals that come from the wild. So they have all the advantages of a wild animal, but they are in a zoo now. And so they can be resampled, they can get checkups. So for example, the jaguar was treating a root canal in its tooth. And so they got a biopsy of the gum, and we got a transcriptome from the gum. And so hopefully animals that are in zoos will be treated in different parts. So we'll get new transcriptomes as time goes by. And then when they finally die in captivity, we're hopefully going to sample it all of tissue for transcriptomes and improve our annotations. So that's high quality reference genomes, which can then be used for population genomic studies using reduced genomic representation. I put in boldface types what I think is going to become more and more the mainstream. This is still done by a lot of groups, but Probably it's going to evolve towards population level, whole genome sequencing. And then for a lot of applied questions, we're going to use those resequenced genomes to design genome-enabled marker panels. There can be SNPs, like in the case of the Jaguars, but other people are developing STRs typed by NGS, not typed in the way we've been typing for 30 years. That was really painful. I don't know if you youngsters have ever typed microsats. We've typed a lot of them, and it's a lot of work. And there are easier ways to type microsats now. But both can be designed based on whole genomes and hopefully build public database with standardized markers and protocols, which will be used for population monitoring, integrated with management in ex situ populations, forensics, and molecular ecology. And we can also diverge from here to look at population genomics, various aspects, and also the population level resequencing for load, local adaptation, other adaptation-related questions. So this is my last slide, just showing what I think is going to happen I hope is going to happen in the field of conservation genomics that has to do with our axis 3, and I can take questions about the other axes as well. Thanks. All right. Sure. Yeah.
The question regards the reference genomes. So for the MCT, do you think you will continue the models that you already worked with for carnivores, or are you going to connect to the biogenome projects and try some of these models that are hybrid between Brazil? I imagine that a lot of this was made in collaboration with other countries.
Right. So I'll just repeat. So Ricardo is asking about the reference genomes and how we're going to deal with that. So for carnivores, we're lucky that there are several reference genomes. So actually, we have not done any reference. Well, we've done the Jaguar reference genome in 2014, but that was a kind of reference genome that's different from current levels, current standards, right? It was Illumina with short libraries and jumping libraries of different sizes. So at the time, that was a reference genome, and we got one. But that's our only reference genome. Everything else we've done is using genomes from other people. So basically, there are dog genome, cat genome, otter genome, lynx genome. And so we're lucky with carnivores that we have, at least within the family, a reference genome for a cat, for dog, for otters. For the coati, we don't. And so that's why I picked the coati for our current project, the one that Camila presented, because this is the first reference genome for this family. I'm also interested in another family, the skunks, the Mephistidae, that has no globally, no reference genome. So I think those two carnivorous families require a reference genome. The otter that we're doing in that project, it will also be useful, but there's at least there are two high quality otter reference genomes. So I think it depends on the question, but definitely we're going to try to follow the models that Camila presented. So we're going to try to either use one or the other. Most likely the hybrid strategy is the one that's going to apply the most, because we have the capabilities to do some of the sequencing. I think it has to do with cost, it has to do with speed. So we're going to try. So the coati is the first try, is the one in the project that Camila described. The otter that Mariana is leading, we're also interested, of course. We're going to use the otter for the next generation of otter projects. And I think both will be done at more or less the same time with a very similar approach. We're just starting to discuss, but most likely a hybrid strategy or the company-based strategy is the quickest. But we understand, and of course, we support the capacity building in Brazil. So if we can, we're going to try to do more steps in Brazil as we go along.
You mentioned that the idea to have this animal from the zoo, but they're originally from the wild. And I know that this is kind of common, but from my understanding, sometimes like it's not official that they take animal from the wild. So my question is like, how common that officially zoo have animals from the wild?
Okay, so the question is about having animals in zoos that come from the wild and how common it is. So in Brazil, actually, it's very common because most of the animals in zoos come from the wild because they were confiscated from the illegal traffic or somebody found them. So there's not a lot of breeding. A lot of species never breed in zoos, or they have a hard time breeding, or they breed sometimes. So most of the specimens in zoos in Brazil come from the wild. Either they are found. So say again? Yeah, the one. Not the exotic ones. Yeah. So the native, especially carnivores, I'm talking about mammals, let's say mammals, but birds as well. Some species breed well in captivity, and then you have the captive bred animals. But a lot of species don't breed that well, or the inflow is large enough because of confiscation, of trafficked animals, or cubs, for example, that are found. They're not trafficked necessarily, but somebody's a rural worker finds a cub. And takes it to a zoo or takes it to a rescue center, or the mother was road killed, or sometimes somebody finds a pup. So you have an inflow. We have rescue centers, we have rehabilitation centers. Some animals cannot be rehabilitated, so they stay and they end up going to a zoo. So that's not a difficulty in terms of some of the species, because most of them, if you look enough, if you look at multiple zoos. So for example, for the quaddy, one of our challenges was to find a male. All the zoos we looked at had females. I don't know why. So it took us months to find a zoo that had a wild, and they do breed in captivity. So we did find eventually one that is a male that was rescued, and it's in a rescue center. And that's the one we're going to sequence.
On the pangenomic approach, how much is it actually used in mammals? Is it too hard to build, and how much advantage does it give to classic approach, like not in the pangenomic way?
Right. So it's about pangenomics and how much is it used in mammals. I don't think it's very much used at all yet, but I think it's starting. So for example, for the maned wolf, we are starting the hologenomic approach, and also looking, we have 3 likely reference genomes to come out in the next year or 2 of maned wolves, 2 from captivity, 1 from the wild. And it's a small pangenome with just 3, of course. But for this fish system that Fred's working on, it's very interesting and very important, because it looks like genomic architecture is a relevant player in speciation in that system. So if all you're doing is the same kind of analysis we've been doing in terms of genomic diversity, runs of homozygosity, structure, I don't think using long reads in the first place is going to change a lot. And it's much more expensive, and you need higher quality DNA. So I think that is our main caveat. What is the advantage is to start looking at adaptive changes in architecture, segmental duplications, inversions, to really look at more sophisticated analysis that take advantage of a much better genome in terms of its assembly, that you can actually look at inverted segments, to look at duplicated segments, paralogs in regions that are really complex, that you can't really assemble reliably with Illumina. Those things we don't really look at most of the time with our current projects. We just look at things that we can reliably infer with Illumina. But I think this is going to open up new questions, and it's already happening in some systems. Of course, in humans, it's happening. It's happening in bacteria. A lot of bacterial work is using pangenomes, and you have a lot of rearrangements. So when you have a lot of rearrangements, and those rearrangements have a fitness implication or some kind of adaptive implication, I think the pangenomic approach will be quite useful, quite powerful. But I think we don't really know yet. We don't know in carnivores how important it is, because we haven't started.
Case of a possible hybridized species, would it have also advantage since it considers a source material different individuals, which may have different levels of hybridization?
Yeah, so in the case of hybrids, I think so. Most likely, maybe some segments, we're already seeing that some segments introgress a lot more than others. So when you have a better assembly, you have long read-based genomes from the hybrid population, probably we're going to start seeing whole segments that don't introgress, or that they get inverted, and that gets kept as a block of genome that is, selectively different from adjacent blocks. So I think all the architectural related questions will be improved when we use pangenomes relative to our current Illumina approach. But as I said, it's early to know. Maybe we're going to find that for some species, it was just more money and we didn't gain a whole lot. And for others, we'll find something completely novel. Next year's we'll say.
Okay, jump here to questions from YouTube, and these are a lot related to the new institute. So first, Lucas is asking if there will be permanent hiring of professionals in genomics in the new institute.
What a good question.
Assembly and annotation of genomes.
Okay, so good question. There will not be a permanent hiring. And the institute, the way the Brazilian CNPq, our council, calls those institutes, unfortunately don't fund the building. Some institutes actually get additional funding and they actually build the building, and they become very few of the 200 or so. Most of them are like a network of institutions that work together and they get funded on a larger scale, but they don't build a physical structure. They use physical structures from the existing institutions. Likewise, all the hired professionals are from the institutions, and what they fund are fellowships. And they do fund. We did get a lot of fellowships in this INCT. We'll be posting fellowships. Where's the camera? We'll be posting fellowship positions, opportunities from the institutes for assembly, for library prep, for bioinformatics, hopefully in the next few months. When we get the actual money, because we haven't gotten the money yet, but it's funded. So hopefully we'll do that soon.
Which also about the new institute, Juliana is asking if it includes plans to sequence reference genomes for ecosystem service-related invertebrates, such as native bees and butterflies.
Yes, absolutely. We're gonna have to choose carefully, right? We're not gonna be able to sequence a lot. Like Camila just explained, what we are gonna do are a few, well, in both the project that she talked about, but also in the institute, we'll not be able to do hundreds. So we'll do 10, 20, a small number of studies that hopefully will foster, understanding of how to do this in Brazil and to improve our pipelines like the way Camila described. So I think there will be a selection process driven by the PIs that are already part of the INCT, but also they can partner with additional PIs and additional groups. So reaching out to the people, you can go back in the YouTube and freeze the screen, and you can see all the PIs that are already in the INCT. So reach out to one of them that you already know and try to convince them to promote your species of interest as a good candidate, because we're going to do at least a few, and that's a good criterion for sure.
Yeah. There is a question about the workshop in population genomics at PUC in Porto Alegre. Is it expected that the applicants have their own data and researchers in the first steps of their project also motivated to apply?
Absolutely. Yeah, you should all apply, and we'll do our best to accommodate as many people as we can. We opened 20 spots presentially in person and 20 online. You do not need your own data. We're going to use a Jaguar data set. It's already been prepared. That's going to be hopefully good enough for all the tools we're going to show. So everybody's going to use the same data set. You don't need to have your own. And we're going to try to contemplate as many people as possible, but we're going to have to select. So people with more experience that already use command line, that already use Linux-based systems will be preferentially selected. People who have already tried to run some of those pipelines, but we'll do our best. And of course, there's a letter of intent there. Try to be very convincing in your letter.
Yeah. And the last question is from Paulo. I already partially responded. If there is a central place to check the network's ongoing and planned sequencing initiatives to avoid overlapping efforts and encourage collaboration.
Mm-hmm.
So I mentioned that there is the GOAT platform, which is a platform for Earthbiogenome project related projects to announce the genomes that they are doing, the ones that are planned, realistically planned, or also in the longer term, so that other projects know about the plans and can reach out, not only on projects, but researchers can go there to this website that I put in the chat and look if the species of your interest is in the planning of any project, which does not mean that it's going to be done. If it's not ongoing, it might just be in this long-term plan. And remember that samples the crucial step. So maybe reaching out to them and seeing whether they would be interested in partnering with you for your species of interest is a good thing. But talking about our network, we will in principle, so we haven't closed the species because since we are in the sampling phase and we don't know if we're going to succeed for all of them, we haven't really announced, but this is going to be in our webpage. And once this is fixed, we will also consider connecting to the EBP and putting the information in GOAT. Yeah, but in our case, you can always go to the Genotropics webpage. This is going to be, I think, I think it's going to be updated in the coming weeks, let's say, by after the July holidays. And then you will know, you can follow there what you're going to be sequencing.
Great. Let me
That's all.
compliment on that.
Yeah.
So as Camila said, there's GOAT, the Genotropics website will definitely have information on all the projects going on with Genotropics scientists and within the networks that Camila described. The Institute will be partners with Genotropics, and actually everybody in Genotropics is part of INCT. Also GBB, we're partnering with them. Alexandre Aleixo, who's the leader of GBB, is part of the INCT as well. And Guilherme Oliveira, the head of the ITV, is on the advisory board. So the goal is that we share information and then we don't duplicate. So we're not going to do the same species. We're going to check with each other before we do each species. So nobody's going to sequence the same species twice unless we want to do multiple individuals. And if people are interested, like Camila said, reach out via the Genotropics mailing list or website, check out GOAT, then try to connect to people and collaborate, because that's how we're going to advance.
Yes, and I think we need to think about a way to gather information at the level of Brazil, because there are many different projects that are working on reference genomes. And for now, there is no way that I know of to know. So we should definitely think about that, so that, we should not duplicate, should find synergies, but at the same time, there is no one rule, because sometimes your goals might not be the same as another project, and timing is different, costs and different types of pressures. So we should have some ground rules and ethics, but flexibility is key, I think, and good scientific practices in general.
Yeah, and also my take is generally we talk to people anyway. So in the case of the carnivores, it's a very popular group and a lot of people are trying to do carnivores. So when I hear that somebody's doing something, I go talk to them, say, wow, are you doing a genome too? Great, let's compare our genome. So let's do 2, let's do 3. Like the maned wolves, for example, we found there are 2 projects going on with reference genomes. We just are starting to discuss the possibility of a 3rd project. So yeah, if we get 3 genomes, they're going to be comparable. And one thing I forgot to say is that the INCT will have a website as well, which is of course going to be connected and linked to the Genotropics website. And so it's a good idea to definitely have some kind of a mechanism to understand, to map out who's interested, who's already doing, and how not to duplicate, similar to GOAT or even connecting to GOAT in terms of what the Brazilian communities do, or Latin American in general for that.
Okay, great. I think that's it from questions. Well, first I would like to thank everyone. First of all, here in person, thank you guys for coming. Everyone watching online, live or afterwards. And I would like to actually invite our colleagues back, especially Ricardo, who didn't present today, but he is part of Genotropics since the beginning and specialized in museomics. Ricardo, would you like to say something to the audience?
No, just that this is a network that is open to everyone. So it's very important to get your feedback. And if you guys are interested in collaborating with us, please join our mailing list. And the same thing for our colleagues watching online. I imagine there's a lot of people in Brazil that are excited with these opportunities, and keep in mind that this project is as big as we are all together. So I hope that many of you can join us in either online or in person. Yeah, that's my message.
Thank you.
And we say our greetings to Fabrício, our 5th member who couldn't be here today with us, but we always in contact with him, and for sure you're going to see him in the next event from Genotropics. Thanks a lot, and bye-bye.
Bye, guys.
Ciao.
Ciao.